From 1989 to 1997, he was on faculty at the University of Guelph, working closely with the Canadian industry on genetic improvement of dairy cattle. He moved to Iowa State University in 1997, where he currently is a distinguished professor in the Department of Animal Science. Current research focuses on the genetic basis of feed efficiency and health in pigs and poultry, and on the integration of quantitative genetics and genomics in breeding programs.
Okay, thank you very much. Thanks for sticking around, and thanks to the organizers for inviting me to talk to you about some of this research. And it's really a very large project that started in 2015, involved a lot of people, including some of the people that are listed here. And I'll talk more about their involvement. Fazir Kayondo was the PhD student who did all the analysis that I'll be sharing, most of the analysis. And then Yolande Seddon, she's an associate professor at the University of Saskatchewan. She's actually in animal welfare, and she did a lot of the project development, but then also the assays of stress hormones in hair that I'll be talking about. Okay, but then it also involved others from the University of Saskatchewan, including John Harding, who is a veterinarian at University of Saskatchewan. Several people from University of Alberta that some of you may be familiar with, especially Mike Dick if you go to the Banff Swine seminar. Mike was actually the lead on the overall project. And then also Frédéric Fortin, who is a researcher at the Centre de Développement du Porc de Quebec, which is the province of Quebec research station for pigs. So yeah, quite a number of universities involved, support from 7 breeding companies that were part of a consortium called PigGen Canada. And all breeding companies that have or used to have a nucleus or multiplier presence in Canada. And the idea behind that consortium was really to set up a research consortium that would help us as academics solicit research funding, especially from Genome Canada, which is the federal government in Canada, but then also a couple of the provincial genome institutes, as well as USDA, and then also another RDAR from Canada, to solicit research funding to answer questions that they couldn't address on their own. Large research questions, pre-competitive research questions, and that's been very successful. Okay, so this is about infectious disease. We've heard about infectious disease already a lot. There's a lot of it out there. For some of them we have good vaccines, many of them we don't. Even if we have good vaccines, new strains appear and the vaccines may not work anymore. As we heard, there may be a solution for PRRS, the gene edit. Okay, but PRRS is not the only disease out there. We have lots of them, and this is only a partial list. And then others that we luckily don't have yet, maybe, hopefully not. But you know, there's a lot on the horizon, and PRRS hopefully is not the exception where we can find a single gene that we can edit that makes the pigs resistant. There may be others like that too, but most likely not, because usually there are lots of factors, genetic factors on the host side involved that determine whether a pathogen can infect that host or not. Okay, and then also the host is changing. These pathogens, they can evolve quickly, more quickly than we can evolve the pig. So I think the end result is that there may be some solutions in terms of vaccines, biosecurity, and so on, or gene edits that may work for some diseases. But I don't think we're ever going to get to a situation where we're not going to have pigs that are not faced with disease, infectious disease. So there's the idea of selecting, and resistance or susceptibility to most infectious diseases is affected by many genes. Maybe there's some larger effects, but generally there are many genes involved. And so the idea here is that we want to breed pigs that are more resilient to disease. So they may get infected, but they are less affected by that infection, right? So they may recover more quickly, or they keep growing. They're less affected by the infection. And I think that's a relevant thing to look at for cases where you can't prevent infection of pigs, which I think is a reality for the foreseeable future. So, for example, if you look at growth rate against disease pressure, 2 pigs, when there's no disease, they have equal growth. When there is disease pressure, increasing disease pressure, the growth rate of pig B is more affected than of pig A, so pig A is more resilient, less affected by the disease for whatever reason. But you can also look at other measures of resilience, the number of veterinary treatments that a pig needs, or mortality, and those are also a couple of the measures of resilience that we looked at in this project. So if you accept that selecting pigs that are more resilient to disease is a good thing to do, then the question is for the breeding companies, well, how do we do that? And this gives the overall structure of the typical breeding pyramid. So the market pigs are usually a 3-way cross, a terminal line, sire line, cross with a crossbred sow. So usually Yorkshire and Landrace. Most of the selection takes place here in these purebred lines in nucleus farms. And they are typically under very high biosecurity, because you need to keep the diseases out of those herds, or you won't be able to sell germplasm out of them. And of course, at the commercial level, you can't afford to have that high of a level of biosecurity. So there's a higher disease pressure. So most of the selection and data recording takes place here, right? So pigs that perform well under high biosecurity, but not all of that translates into higher performance at the commercial level, where there is some disease, depending on the farm. Okay, so then the question is, well, how should breeding companies select pigs that are more resilient? Because if you want to measure resilience, you have to expose them to disease, and they can't do that at the nucleus level. So now there are different ways to do that. You could collect data at the commercial level and then genotype them, and then identify the genes or the markers that pigs that are more resilient at this level have, and then select for that. That's possible, and some companies do that, but that's very expensive to collect that data here and to connect them back to the nucleus level. It's very expensive, but it's possible, and it's been done. The other alternative, and that's what specifically we looked at, is using what are called indicator traits. So are there things you can measure here on these purebred animals without disease that are good genetic indicators of whether their progeny are more or less resilient when they come to a farm that has some disease pressure? Okay, maybe some blood test that you could do. And in this case, what we're going to looking at is levels of stress hormones in hair. So it has to be, in order for you to be able to have an indicator trait for disease resilience that you can measure here, it has to be measurable on healthy pigs, because you have to keep these pigs healthy. Preferably you do that, you can collect that at a young age. No blood sample would work. It has to be heritable, because you select then based on that indicator trait here, and then you're hoping that they have the better genetics for resilience, even though they don't express it because you don't have much disease here, but they will pass those genes on to the commercial level. Okay, and you get improvement there. So they have to be heritable, and they have to be genetically correlated with disease resilience. So the indicator traits, like a blood test, right? So you're not directly measuring disease resilience, but you're measuring something that, based on the genetic correlation, you can quantify to what extent that indicator trait is affected by the same genes or some of the same genes as disease resilience, right? So so you need both the indicator trait to be heritable and to be genetically correlated with disease resilience. So finding these indicator traits was a big part of this project, this large-scale project, the Natural Disease Challenge Model project, that was started in 2015 in this research barn in Quebec of CDPQ, located just north of the St. Lawrence Seaway. So Quebec is here, New York is here. Beautiful location. They had a nursery quarantine site, and then this is the site where they introduced diseases into a barn, into a nursery finish barn. And they did that by bringing pigs from farms that had disease problems in their neighborhood. So they brought sick pigs into this barn, and let's see, and the idea was to create high disease pressure, even much more than you would see in a typical commercial farm, because we wanted to have a lot of expression of disease resilience. So we was set up to have multiple pathogens present in that barn, and very high disease pressure, a lot of veterinary control to make sure that it doesn't get out of hand, but also that we maintain most of these pathogens in the barn. And the way it was maintained was by continuous flow. So rather than every batch introducing new cedar pigs, it was maintained by continuous flow, where a new batch of 60 or 75 pigs was introduced every 3 weeks. And they were introduced initially into this quarantine nursery. They stayed there for about 28 days or 21 days. And then they were moved to this barn, into the nursery of this barn, where they, for 1 week, they had contact, contact with the previous batch, right? So the older batch passed by horizontal transmission, passed the pathogens on to the new batch. So we did what we tell none of you to do, which is continuous flow. But that's the way the pathogen load or the disease pressure was maintained from batch to batch. And this was kept up for 7 years. And in total, 4,700 York landrace barrows that were sent through this system. Each batch came from one of the 7 breeding companies, from one of the 7, a multiplier from one of the 7 breeding companies. Of course, the barrows they don't need from the maternal barrows, so they're a byproduct, and so they could go to this project. And each batch is from one multiplier, from one company. The idea here was not to compare disease resilience of different companies. And we can't, because each batch will be exposed to its own set of, its own level of disease, right? There's lots of things changing from one batch to another batch. But that wasn't the point either. It wasn't the point to compare breeding companies. We wanted to look at the genetics of pigs, the difference in genetics and disease resilience of pigs from the same company or within a company. Okay, so these 60 or 75 pigs from that batch, from that company, they were then followed, and lots of things were measured once they got to this disease challenge barn. growth performance, feed intake and behavior, drinking behavior, health treatments, mortality, carcass traits. It's a research barn, so they have individual feed intake recording with RFID tags in the finisher, and also they had drinking water intake recording in the finisher, and then also in some of the later batches they also had feed intake and water intake recording in the nursery, in the late nursery, okay, using the Acerva system, or Pig Insight it's called. So lots of data being collected on those pigs. And then we also, we collected individual health treatments and mortality. This is an example of one of the batches, the growth, body weight over time growth. The red ones are the ones that died. On average, about 25 of the pigs died. Okay, and that was sort of the target, this mortality rate that was targeted. So huge mortality. They died for all kinds of reasons, many because of respiratory. There were multiple strains of PRRS in there, and flu. Also diarrhea was a heavy reason for mortality. So that was recorded on all the pigs that died. And then also there were group treatments, but we were really interested in the individual health treatments that were applied to pigs. And some pigs didn't require any treatment, others 10 or more, right? So a lot of variability, and that was all recorded on these 4,700 pigs. Okay, and then for genetic analysis, they were all genotyped using 650,000 genetic markers across the genome. So we could scan the whole genome for which traits contribute to all of these disease resilience traits. I mentioned veterinary oversight in animal care, huge part of this project, to be able to expose them to this severe disease. Very dynamic. This is weekly mortality rate over some period, varied a lot. And also different management steps were taken to control, to make sure that if the mortality got too low, that we increase the severity of the disease, or if it's got too high, to slow it down. So for example here, stop nose-to-nose contact when the new batch enters the disease challenge. So rather than putting them in pens next to the old batch, they were put in opposite sides of the barn. So they were still in the same barn, in the same airspace, but they didn't have nose-to-nose contact, and that tempered how hot the challenge was. But then at some point that would be started again. Sometimes one of the strains of PRRS or flu was not there anymore and was reintroduced. So all kinds of things happened. So this disease challenge was very dynamic from batch to batch. So many of you will ask, well, you're never going to get reliable data from this. You know, we're used to controlled infection experiments where we inject pigs, infect them with a given dose of the pathogen, and then we do the collect data. This is very dynamic, but it's also real life, right? Also in a commercial farm, it's very dynamic. So first question, well, does it generate reliable data? And one way we could assess that is by looking at whether the traits we're recording are heritable. If we're collecting average daily gain, for example, in this system, and it's such a noisy system that, very dynamic and lots of things changing, we're not going to get high heritability, right? Because most of it is just, environmental. So Austin Putz, who now was a PhD student, now works for Hypor, and Mark Cheng was also a PhD student and now works for Bayer Plant Genetics. They looked at some of that data and they found that for many of the typical traits, heritabilities were very similar to what we see in healthy farms, right? Growth rate. About 30% in the finisher, about 30% of the variation, the differences in phenotype are due to genetics, and that's similar to what we see in healthy barns. It doesn't mean it's the same genetics. And we've actually estimated genetic correlation between growth rate in this under the challenge with growth rate in a healthy environment in the nucleus, and it's about 0.5. So it says that only half of the genetics that is important for growth rate in the nucleus is also important for growth rate in the commercial level, and the other 0.5% is different. Okay, so they are different traits but similar heritability. And then for the health traits, typically low heritability, but higher than we observe in literature, and that's because of our disease pressure was fairly high, so ranging from up to 15% heritable. So pretty reasonable heritability. Some of them weren't heritable. The number of treatments in the finisher was not heritable, for example, but and you can imagine that the reason for treatments or the reason for mortality may not be very heritable. Okay, so that's the whole system. One of the things that we did is in the quarantine nursery, and they were there for about 3 weeks. So they arrived from healthy multiplier farms and then went into the quarantine nursery. We collected a lot of things to try to find these indicator traits. So traits that we can collect on healthy young pigs, and that, and as I mentioned, they have to be heritable, and they also have to be genetically correlated with things that we measure here, the resilience phenotypes. So we've published a lot on that. I'm not going to talk about those. There's some promising results there. What I'm going to talk about are stress hormones in hair. So stress, we're all exposed to it. There's all kinds of different types of stress. It invokes the HPA axis, or the hypothalamus pituitary adrenal gland axis, producing glucocorticoids. Cortisol is the most famous stress hormone, right? So, but then there's also DHEA, which is sort of the antagonist of cortisol. And then there are the inactive forms of those two, and all 4 of them we measured. So they get produced under stress, and they get released to the bloodstream, and they make it to a hair follicle. And then as the hair grows, these hormones get deposited in hair. Right? So what you can then do is you can collect this hair and then measure it for those hormones, and it will tell you something about the stress response that the individual had over the time that this hair was grown, right? You could even, if some of you have long hair, you could look at the tips of your hair, right? Just look at the tip, and it tells you about the stress hormones that you produced when maybe several months ago, right? We did not do that. We actually cut the hair twice, so we had the regrowth to look at two different stages. So in pigs, they also get exposed to a lot of stress. If you think of the disease challenge model we had, even before they get into the disease challenge, they're exposed to being weaned, being mixed, being processed, transportation. So there's lots of stressors there. And then once they get to the disease challenge, of course, disease is a huge stressor, right? And so when it comes to disease, both if you're healthy or sick, it also invokes the stress response, and vice versa. If you're sick or if you're stressed, you're more likely to get sick, right? So the immune system and the stress response system, they're pretty closely tied. So what we did is we cut the hair twice, and we shaved the hair at the back of the pig here twice. Once just when they went into the challenge nursery, so we, if we had to let the hair grow, it would have been the tip of the hair, and then we once they about 40 days into the disease challenge, we cut it again, right? The regrowth. So the idea is that this hair, if we measure stress hormones in this hair, it's mostly response to non-infectious stressors because they have not been exposed to major diseases, right? whereas this hair here is mostly infectious stressors, right? Huge disease stress. And all those essays, they were done in Yolande Seddon's lab at the University of Saskatchewan. We also did a back test, which is a standard test to evaluate coping styles of pigs. Basically putting the pig, and it was done in the quarantine nursery, basically putting the pig on his back, and then for 30 seconds count the number of vocalizations, the number of struggles, and the intensity of those. Okay, I'm not going to say a whole lot about that, but so first thing we need to know. So the question was, well, can we use, especially this haircut? We can collect that on young, healthy animals, right? Can that be a good source of an indicator trait? So stress hormone levels measured in hair collected at that point, does that give us information about disease resilience? Okay, genetics of disease resilience. Well, for that they have to be heritable, right? And the blue lines here are for hormone levels in hair cut before they go into the challenge. The other bars are when they during the regrowth during the challenge. And you can see for some of these are not heritable, but especially cortisol was heritable at in both samples. Okay, reasonable heritability. I'm not going to go too much into this. Well, okay, one thing that I want to show here is that the genetic correlation between the cortisol level in this hair versus this hair was also only 50%. Okay, so there is different genetics that controls the cortisol level at this stage, right? The non-infectious stressors versus in hair that's grown under disease stressor. Okay, so there may not be fully the same genetics. I'm going to skip the back test responses for now. Yeah. We did so we had genome markers across the genome, so we can do what's called a genome-wide association study. So when we look at cortisol level in hair in one of those samples, we can see, well, which parts of the genome cause genetic differences in cortisol level. And what we found, this is in the nursery and this quarantine nursery and the challenge nursery, what we found is that there was a huge region on chromosome 2 that explained over 40% of the genetic differences in the quarantine nursery sample, and over 35% of the genetic variation in the disease challenge sample. Huge effects. And lo and behold, that's right in the region where the glucocorticoid receptor gene is located. That's the gene that cortisol binds to have its effect. Okay, so biologically that makes a lot of sense. Let's see. And we also found, this is again this genetic correlation between cortisol at the two levels. We found that this gene explains most of the genetic correlation. Right? So if we take out the effect of this gene, the rest of the genetics that affects cortisol here versus here is affected by different genes. Okay? So that was also quite interesting. Then we looked at, well, what's the effect of that minor? So is that, then you can ask, well, is that an allele that we want to select for, right? So, well, first of all, this is what it does to cortisol. So first of all, this allele, the major gene, the frequency of the allele that decreases cortisol was only 9%. So most of the pigs were actually had the allele, the copy or the version of that gene that increased cortisol. If they have one copy of the decreasing allele, it decreased cortisol by 30%, which is a huge effect. Okay, and also cortisone, it reduced by about 25%. So, indicating that pigs that have the what we call the minor allele, the decreasing allele, have much lower cortisol levels in hair, perhaps indicating that they're less susceptible to stress. Okay, let's see. So then we wanted to know, well, are they good genetic indicators? So it has to be heritable. We already know that they're heritable. Are they genetically associated with resilience? And we can then look at both at this major gene. That's something we can select for, right? If the decreasing allele is also increasing resilience, then that's a good thing to select for. And then also we can look at the genetic correlation between hair hormone levels and resilience. So for that we use the disease resilience traits that we collected on all of those animals. In total we had, there's about almost 900 that had the stress hormone data, but we had over 4,000 that had the disease resilience data. They all come from the same companies. This is the effects of the minor allele at the major gene for the cortisol on disease resilience. And so, for example, what we found is that the allele that decreases cortisol in hair increases feed intake under disease, which is probably a good thing, right? It maybe increased FCR, but you want pigs to eat when they're under disease. It didn't really translate in significant effects on growth rate, but standard errors are large, so we're affected by the limited number of animals. But this is probably a good thing. Then when we look at mortality went down. So pigs that had the allele that decreased cortisol had less mortality, at least in the finisher and across the finisher and the nursery. It didn't really affect, there was no clear signal for health treatments, right? This is interesting. So the pigs that have the allele that decreases cortisol, they have lower carcass weight, they have lower dressing percentage, and they have they're less lean. Does this sound familiar? Anybody know of another gene that behaves in the same way, that increases susceptibility to stress and increases leanness? Goes back to a person that we talked about this morning, Lauren Christian. He was one of the people that I've identified the stress gene, the halothane gene. So the halothane gene, if you have the unfavorable effect, you have much higher cortisol levels under stress, and you also have much higher leanness. And that's how the halothane gene was increased in frequency, because breeders were starting to select for leanness against back fat, and then they indirectly selected for the stress gene. Right? Well, this is the same way. So here is it's sort of the opposite, but here I'm showing the allele that decreases cortisol, also decreases leanness. Right? So if you select for leanness, you're selecting for the allele that increases cortisol. Very much aligned with what we found for the halothane gene. The halothane gene was on the chromosome 6. This one is on chromosome 2, so they're different things. Also, the mechanism was different, because the halothane gene was the ryanodine receptor, which was associated with calcium transport. This seems to be more directly with the glucocorticoid receptor, but very similar thing. So you could say, well, maybe we don't want to select for that. But I think that still this is a good candidate allele to select for, because if you select for the allele that decreases cortisol, you're going to increase feed intake under disease, which is good, I think, and decrease mortality. Maybe you have lower leanness, but we have other tools to compensate for that. We have good genetic evaluation for leanness, so we can compensate for that. So I still think this is a good thing to select for. When it comes to genetic correlations, so it becomes a little fuzzier, and part of it is because the we don't have enough data. These standard errors are large, but if I kind of go through this, if you look at the growth rate traits, so here's growth rate and feed intake, the genetic correlations between cortisol level, the blue bars, and the growth rates, they tend to be negative. In other words, pigs that have higher levels of cortisol have lower growth rate. So again, you want to select for lower cortisol. Okay, same as the direction for the one major gene that we found. This is cortisol in the challenge nursery, in hare challenge nursery, and some other correlations. And I'm going to have to wrap up soon. Here also we see that same antagonistic relationship with leanness. Okay, pigs that have higher cortisol also have better leanness, right? And some mortality goes in the expected direction. So the pigs that have higher cortisol have more mortality. So you want to select for lower cortisol, and some of the other. Unfortunately, in the quarantine nursery, it's much less clear. Okay, so it doesn't look like, apart from that major gene that we found, it doesn't look like the other genetics affecting cortisol collected from hair from young healthy pigs may not be all that informative. But standard errors are huge, and so we really need to get more data, really, to get the bottom to this. But even this would be useful, because then breeding companies could go to commercial farms that have high disease pressure and collect hair on pigs from commercial farms, right, which would kind of reflect hair grown under disease, which is what we have here. And that may be good information for them to then use for genetic evaluation. Okay, I'm skip through this. Okay, so summary, so levels of stress hormones in hair are moderately heritable, and they're basically retroactive measures of stress response, because you're evaluating the level of stress hormones that you had in your blood system as the hair that you're analyzing was growing, which could be months, right? And there is some genetic correlation. It's mostly driven by that major gene between the hair from healthy pigs and hair grown during disease. We found that major gene, and that's certainly a good candidate for selection. And then also the hair, stress hormones in hair from pigs that have been exposed to disease can also serve as phenotypes for to evaluate genetics of disease resilience, because it is a phenotype that's more heritable than mortality is, or that treatment rates are. And I didn't talk about the Beck's test, but it's also a potential target. And so then I want to finish up again by thanking everybody, all the funders, all the collaborators, and thank you for your attention.
Are there any quick questions for Jack?
Yeah, I'm curious about the relationship between leanness and the cortisol level. Did you look at the genetic correlation between leanness and the resilience traits?
And between leanness and?
Between leanness and your resilience traits, like mortality and treatments.
Can you restate the question?
Okay. Question is about the relationship between leanness and resilience, right? Whether we looked at genetic correlations between leanness and mortality, and I don't think we have. So I don't know.
I can say.
Yeah.
So this is more like a chronic stress. Is still there any merit in also measuring acute stress at some at the moment they start, or this just with doing 2 hairs collections is enough, you think?
Yeah, that's a good question. So yeah, we really wanted to focus on chronic, right? Because we are looking at the response in that first hair sample responds to all the stressors that they've been exposed to over that time period. And then the second hair sample is all the disease pressure they've been exposed to during that 40-day period. But yeah, so that's one measure of resilience, right? One target for resilience. But yeah, you could also look at acute response stress, right? And people do that by giving them an ACTH injection to stimulate that glucocorticoid response and see how quickly it goes, up and down. And so that's another target, right?
Do you know if they are genetically correlated or no data?
I don't know whether those are genetically correlated. No. From my standpoint, I think the chronic one sort of averages things out across lots of different stressors and lots of different pressures of disease and stages of disease. Yeah.
Tim Ramsey once found that Dr. Tim Ramsey investigated the association of stress and acute phase proteins, the accumulation of acute phase proteins. Do you think there might be a correlation with that as well?
There could very well be. I mean, acute phase proteins, they've also been shown to be related to resistance, resilience, or immune response, and we haven't looked at that. But that's a good suggestion. Yeah. We still have all these blood samples are in storage, so we still have the opportunity to go back, and I think that's a good suggestion.
All right, now that we're a little over time, if there's any other questions, you can ask privately after we wrap up. But can we get a round of applause for Dr. Dekkers?